Mitosis is used for almost all of your body's cell division needs. It adds new cells during development and replaces old and worn-out cells throughout your life. The goal of mitosis is to produce daughter cells that are genetically identical to their mothers, with not a single chromosome more or less. Meiosis, on the other hand, is used for just one purpose in the human body: the production of gametes, sex cells, or sperm and eggs. Its goal is to make daughter cells with exactly half as many chromosomes as a starting cell. To put that another way, meiosis in humans is a division process that takes us from a diploid cell, one with two sets of chromosomes, to haploid cells, ones with a single set of chromosomes. In humans, the haploid cells made in meiosis are sperm and eggs. When a sperm and an egg join in fertilization, the two haploid sets of chromosomes form a complete diploid set, a new genome. In many ways, meiosis is a lot like mitosis. The cell goes through similar stages and uses similar strategies to organize and separate chromosomes. In meiosis, however, the cell has a more complex task. It still needs to separate sister chromatids, as in mitosis, but it must also separate homologous chromosomes, the similar but not identical chromosome pairs an organism receives from its two parents. These goals are accomplished in meiosis using a two-step division process. Homologous pairs separate during the first round of cell division, called meiosis 1. Sister chromatids separate during the second round, called meiosis 2. During the G1 phase, metabolic changes prepare the cell for division. At a certain point, the restriction point, the cell is committed to division and moves into the S phase. During the S phase, DNA synthesis replicates the genetic material. Each chromosome now consists of two sister chromatids. Then moves on to the G2 phase. During the G2 phase, the cell double-checks the duplicated chromosomes for error, making any needed changes. And once interphase is complete, prophase 1 begins. In prophase 1, the homologous chromosomes pair and exchange DNA to form recombinant chromosomes. Then metaphase 1 begins. After crossing over, the spindle begins to capture chromosomes and move them towards the center of the cell. Each chromosome attaches to microtubules from just one pole of the spindle, and the two homologous of a pair bind to microtubules from opposite poles. So during metaphase 1, homologous pairs, not individual chromosomes, line up at the metaphase plate for separation. In anaphase 1, the homologous are pulled apart and move apart to opposite ends of the cell. The sister chromatids of each chromosome, however, remain attached to one another and don't come apart. In telophase 1, the chromosomes arrive at the opposite poles of the cell. Cytokinesis usually occurs at the same time as telophase 1, forming two haploid daughter cells. Well, that's all meiosis 1. Now meiosis 2 begins. In meiosis 2, the sister chromatids separate, making haploid cells with non-duplicated chromosomes. During prophase 2, chromosomes condense and the nuclear envelope breaks down if needed. The centrosomes move apart, the spindle forms between them. In metaphase 2, the spindle microtubules begin to capture chromosomes. The two sister chromatids of each chromosome are captured by microtubules from opposite spindle poles, and the chromosomes line up. In anaphase 2, sister chromatids separate and are pulled toward opposite poles of the cell. In telophase 2, nuclear membranes form around each set of chromosomes, and the chromosomes decondense. Cytokinesis 2 splits the chromosome sets into new cells, forming the final products of meiosis. 4 haploid cells in which each chromosome has just one chromatid. In humans, the products of meiosis are sperm or egg cells.